What we'll start looking at today is protein synthesis. So we've looked at it sort of a little bit before, but a little bit differently. So to look at protein synthesis, so it's biology, look at protein synthesis, start that today, and probably actually start the first process of it today, do the second part of it tomorrow, and then maybe recap it all on Thursday and then spend all of next week maybe doing chemistry, or I'll just sort of see how long it takes us. But yeah, so I'll start off with gene expression and protein synthesis. So I'm just going to start off with basically, I think I'm on the wrong page actually, to be honest. Yes, I am. It's not helpful. Protein synthesis, that is transcription, which is the first stage protein synthesis, which I want in a bit. Here we go. So basically what we're going to look at is how proteins are made, so obviously synthesis, meaning the making, the production of something, so proteins, so the making of proteins, and it's basically split up into two parts. We call the parts transcription and translation. And we're going to look at the first section, which is transcription today. So we're going to look at how proteins are synthesized from DNA. So how do we basically go from a genetic code to actually fully formed 3D proteins? Describe that genetic transcription. So what do we mean by it? What happens? And then how some hormones can affect gene expression in a cell. Really, we're going to sort of focus more just on the transcription part today. So, gene expression. What do we mean by gene expression? So we know that basically we've got our DNA, and on our string of DNA, like here, our DNA is double-stranded. So we've got two strands of this complementary genetic code. And this genetic code, somewhere along the DNA, we have areas that are called genes. So some areas are coding, which means they code for proteins, and some areas are non-coding, which means they don't actually code for anything. And there's a lot more to it. Years ago, I say years ago, probably about 10, 15 years ago, probably about 15 years ago, we would say, oh, it's just like junk DNA. We don't really know what it does. Like we have these areas that code for DNA, that code for proteins, they're genes, we know what we do, and we just have all this other junk. Actually, we will look into that at some point. This junk isn't really junk, it does other stuff. So these non-coding areas of DNA do actually have other roles. But if we think all our DNA in every single cell is identical, so the DNA that you find in your skin cells, if we extracted that, would be exactly the same as the DNA you find in your hair cells or your liver cells. Now, your DNA is what controls your cells and tells your cells, well, you're going to be this, you're going to be that, you're going to be a liver cell. It controls your skin cells, tell it be a skin cell. So basically, it does this by genes. So not only do we have this big long strip of DNA and just parts of it are genes, we don't actually need all our genes to be working all the time. So the genes for something being, let's think about muscle cells, for example, able to be stretchy, able to move, have lots of, so genes for that, those sort of characteristics need to be switched on. Whereas stretchy genes, and it's calling them stretchy genes, but there will be genes that allow them to move in that sort of way, aren't going to be switched on in your bones, for example, because that is the opposite characteristic. We don't want our bones to be stretchy in that way. So on our DNA, we have genes that are switched on and genes that are switched off in different parts of our body. So that is why all of our cells contain the same DNA, but they can look and work differently. So we can switch genes on and we can switch genes off. So we call them switched on, or we can say they're expressed. So we're expressing that gene, but we're not expressing this gene. So for example, you've got genes for eye color. all your DNA, no matter where in your body, has a gene for eye color. Those genes are only going to be switched on or expressed in your eye cells. They're not going to be working, for example, switched on in your liver, because that is of no use to your liver whatsoever. So when we switch on these genes or express them, obviously the DNA actually has to do something. So the whole point of DNA is it makes proteins. And then these proteins go on and tell our body what to do. So when a gene is switched on or expressed, it's transcribed into mRNA. And mRNA is then translated into a protein. So this is that two-step process. So basically, our DNA has to become this molecule called mRNA, and then it can become a protein. So it's a three-step process, basically. So we go from having DNA, we go to mRNA, and then we go to our protein. Now, we can't go straight from DNA to a protein, and that's because of where proteins are made. If you think about your cells, back to basic cell knowledge, we know proteins are synthesized in ribosomes. Ribosomes are attached to the Golgi apparatus, and the Golgi and the ribosomes are obviously found in the cytoplasm of the cell. DNA, as you know, is found in the nucleus. DNA can't leave the nucleus. That's really good. Basically, DNA is really, really fragile and it can break down. So we keep it inside the nucleus. We keep it nice and safe. It keeps its integrity, means it doesn't get damaged, means it can't mutate as easily. But if we want to make proteins, well, they're made outside the nucleus. So what do? We go through transcription, which is our first stage of protein synthesis, which is where DNA becomes mRNA. And so this stage is called transcription. And that's basically turning our DNA into mRNA so that it can leave the nucleus. So it's basically like a copy. And I'll talk about that a bit more detail in a minute. But it's an RNA copy. And that's just made because RNA can leave the nucleus. So and the M in mRNA, the M stands for messenger, which probably makes sense because it's working like a messenger, taking that information from one place to another. And this messenger RNA is able to leave the nucleus, and it then is able to go to ribosomes, which then our protein can be made and synthesized, and that is a section called translation. So we're translating it from one type of molecule to make another. That's the basics of it. We're going to focus on this first stage today, which is transcription. So quick recap while we're here. Let me get rid of all that, erase all drawings. RNA. So we know DNA is a double-stranded molecule. It's got that sugar phosphate backbone and nucleotide bases. RNA, whether it's messenger RNA or any other types of RNA, which we will come across later, we know is single-stranded, so it's not double-stranded, so it's good to remember this. So RNA is single-stranded. It doesn't have a deoxyribose sugar, it has a ribose sugar, hence it being called RNA. And remember, when we talk about bases, we have uracil present, not thymine. So that is good to remember as well. And we'll look into that in a bit more detail in a moment. So there are key points about RNA. So let's get started then. So genes are switched on. The ones that are switched on in that cell, we transcribe them into RNA and then translate them into proteins. The reasons that we switch genes on and off is because basically we don't want to waste loads of effort, loads of energy making all these proteins that we don't need. So for example, one that helps control eye color, if we make it in our liver, is completely useless. So that'll just be a waste of energy. So proteins, which is what we're going to make from our DNA. So proteins can be things such as enzymes. they can control the chemical reactions, so therefore determine how an organism grows, develops, and functions. So this is that idea again about the eye colour. It doesn't make our eyes blue, it makes a protein, which might be an enzyme, which can control the chemical reactions that are occurring in your eyes or in your eye cells, and it determines how an organism grows, develops, functions, same as how it's controlling how your cells develop, how they function. Purpose of it. So again, like I said earlier, only some of the many genes in the cell will be expressed at one time. And this is really important in controlling development and cell differentiation. And actually, it's not always the same cells, same genes that are expressed over time. So if we think to embryos, they're going to be expressing completely different embryonic cells, and be expressing completely different genes to what adult cells are going to be expressing. Same DNA, that DNA is not different. They're going to express completely different sets of genes, but sets of genes that possibly aren't expressed ever again, and vice versa. Lots of them that we need to be expressed a lot further down the line when we've got a fetus or we've got an actually fully grown adult are not going to be used until those stages. So it's very, very complex. But at the minute, we don't need to worry about when the genes are expressed, other than knowing that they're not all expressed at the same time and they can change. But actually, that when we're making the proteins, it's only the expressed genes. So, oh, what's happened here? Okay, so genetic code. Genetic code's really interesting, actually. We've talked about genetic code before, but I'm going to just do a bit of a quick recap now with you. Really important. So our genetic code, if we think about it, is our DNA. And we've got a few bits of information about it. So we say that it is almost universal. And what we mean by that is the same sequences of bases code for the same amino acids in all organisms. Now, if we're thinking about amino acids, remember that basically what happens is our DNA are nucleotide bases. Okay, so we've got all those rows and rows of bases. So we've got A, T, C, G, U, or U for RNA, so A, T, C, G, all different orders. And basically these bases are going to code for amino acids, which makes sense because obviously once we've got a chain of amino acids that can fold and form into our protein. And it's almost universal. So almost the same sequences of bases code for the same amino acids in all organisms. And we say almost universal because there's a few little differences in a couple of organisms, but nothing you need to know about now. But basically we can actually say it's universal, but technically it's almost universal. There's just a few tiny differences occasionally. And we have this thing called a triplet code. And basically what we mean by a triplet code is all that rows and rows and rows of those long lists of letters, all our bases in our DNA. We split them into triplets. We split them into a group of 3. And an amino acid is coded for by 3 bases. So ACG might code for 1 amino acid. ATT codes for a different amino acid, and it works in groups of 3. And the reason that we have it in groups of 3 is because of the amount of amino acids we have. We only have 4 bases, and we've got more than 4 amino acids. So obviously we can't just have it as 4 bases, code 1 base codes for 1 amino acid. It doesn't work. So they're in a triplet. So triplet code means that we've got 3 nucleotide bases. And when we have 3 together, we call it a codon. And these code for 1 specific amino acid. So yeah, we call these a codon. Those 3 together, we call it a codon. It's non-overlapping. What we mean by this is each base is only part of 1 codon, and each codon codes for 1 amino acid. So I'm going to show you what I mean by that now. So if this was our DNA, got A, C, G, T, T, A, C. So we could split these into triplet codons. So this would be our first triplet codon, ACG. This would be our second, TTA. So that's a triplet codon. So that's one codon. That's the second codon. It's non-overlapping because each three are just are only found in one codon, and we don't use them for more than one. If it was overlapping, it could look like this. But that's not the case. So it's non-overlapping. And then we have this last little bit. Where's it gone? The coded degenerate is a really strange use of the word degenerate, and it often confuses people. But then once we've learned it, we're like, oh yeah, I know what this means. And basically, the degenerate means, I'll join it up, a given amino acid may be coded for by more than one codon. And basically, I will talk it through now what we mean by that. So let's go back to here. So let's move that out of the way. So triplet codon. So our bases, we've got C, A, G, and T. They code for amino acids, and they code in that triplet codon. So 3 together make a triplet codon. It's non-overlapping, as we've just said up here. We've looked at it. It's non-overlapping, and it's what we call degenerate. So it's non-overlapping, and it's degenerate. So what we mean by this is we've got 64 possible 3-letter combinations. And we can see this basically, we've got 4 letters, and the way that you could combine them means that we have got 64 different possible 3-letter combinations. So I'm going to pop that on here. So we've got 64 3-letter combinations. Obviously, we don't have 64 amino acids. So what we mean by it being degenerate is the fact that actually more than one triplet can code for one amino acid. So I'm going to show you now an amino acid code table, and I'll show you what I mean by that. Triplet codon. They're all exactly the same. People just make them look slightly different sometimes. Here we go. So here's one. And basically, as we look at this, we can see here that we've got, for example, for leucine here, we've got 4 different triplet codons that can code for that. For glycine, we've got 4 different ones here. Arginine, we've got 1, 2, 3, 4, 5, 6 different triplets that can code for it. So that's what we mean by degenerate. So given an amino acid may be coded for by more than one codon. So protein synthesis. How do we actually get to making these proteins? So protein synthesis involves the production of a chain of amino acids that form that primary structure of a protein. And this sequence of amino acids are coded for by a gene. That's the basic concept. We start off with transcription. Now, transcription of the gene happens in the nucleus, and that's basically transcribing from the DNA an mRNA strand. And that means that that strand can then leave the nucleus and then go to translation. And translation is where we're actually then going to make that polypeptide chain. And so the mRNA is translated to ribosome to form a polypeptide chain. And then we can modify the protein for our final step. So transcription. This is the key point. This is the first bit. We're going to take our DNA and we're going to turn it into mRNA. How does this happen? So this is what we're going to spend our time looking at now. I'm going to go through it a few times in a few different ways. I normally shorten it, can sort of get it down to 6 steps, but lots of people sort of do it in different amounts of steps. So I think this one might do it in different amounts of steps, but don't worry too much. So basically, we've got our DNA. We've got a double-strand of DNA like we've got here. Okay. Oh, I can pause it. To make a copy of this, if we think back to DNA replication, which we did ages ago, you had to unwind that DNA to copy it to make an exact copy for mitosis. Transcription isn't the same as DNA replication, but the beginning has that same idea, is the fact that we need to unwind the DNA. We can't do anything with the DNA whilst it's basically bonded together, and we've got that double helix. That double helix is great because it means that we can fit it into a smaller space, and it also provides a lot of safety and security for it. It means it's less likely to get broken down and damaged. So we need to actually break those bonds. We need to break those hydrogen bonds between the bases on both of the 2 different strands. So what happens is an enzyme called helicase comes along, DNA helicase. And what DNA helicase does is it breaks the bonds between the double-stranded DNA. Now, it doesn't just do this at the beginning of the DNA strand. That would be really, really pointless, because then we would unravel the whole DNA strand and we would just have a mess of DNA absolutely everywhere. So what we basically actually do is the DNA helicase attaches at a specific region, and we call these promoter regions, and they will have a start codon. That's a specific codon telling the DNA, the helicase enzyme, where to attach and where to start basically breaking these bonds apart. And because we don't want just this whole mush of DNA floating about the place. Now, this is where I'm going to argue with this. So I've said enzyme helicase here. It can be helicase. It can also be a different enzyme. There is 2 enzymes that it can be. It can be helicase. It can also be RNA polymerase, depending on which exam board you're doing and where you look. I would say personally, and everything else I'm going to do with this, I'm going to call it RNA polymerase because that's what I've always called it. And that's what I've got written down absolutely everywhere. That's what I was just doing. I was just double checking everything that I have ever done. And I'm like, yeah, no, it's RNA polymerase. So yeah, I'm going to cross that out. Let's see if I can do that on here. Annotate and draw. Yeah. So I'm going to call that RNA. No, not helicase. RNA polymerase. I'm going to cross that out and I'm going to call that RNAP. So RNA polymerase is going to break down those hydrogen bonds. Helicase does do exactly the same thing, but helicase we use in DNA replication. RNA polymerase we use in transcription. The RNA polymerase is going to come along. It's going to break those hydrogen bonds between the bases. And again, that's in a specific region of double-stranded DNA. It is not, and we call these areas the promoter regions. And they are going to have a start codon telling it to go there. It's not just random. It's not just like, oh yeah, I'm just going to start here. It has a specific reason for starting in a specific place. It's got a start codon telling it to do that. And it's only of those express genes, remember, that are actually switched on. So there's quite a lot of things telling it where to attach and where not to. So that's our first stage. RNA polymerase comes along, it attaches to our DNA, and our hydrogen bonds break. Our DNA double helix starts to unwind, and we have now got 2 strands. Now, I said it's at the promoter region. So what will actually happen is we get, like when we were doing DNA replication, we have a replication fork. It's similar to that. We don't unwind the whole strand of DNA. We just unwind it at certain points where these promoter regions are, and it will keep going and keep going and keep going until it gets to a stop codon, and then it will fall off. Because we don't want to unwind the whole DNA, because that would just be pointless, because we don't need to transcribe the whole DNA. So we just do it in certain areas. So the 2 strands separate. So the DNA helix unwinds, but we don't have 2 fully separate strands, but they're separated in an area. So that is the first bit. Step 2. I am going to use something else because I'm going to fall out with this. Yeah, no, I'm going to fall out with this quite quickly. That's replication, which is not helpful. Here we go. Here's transcription. I'm going to draw it for you. So let's clear this. Can I clear it? There we go. Clear canvas. Get rid of that. Make sure I'm showing the right screen. Here we go. All right, I'm going to draw it myself because I'm falling out with that other one. So we are going to do transcription. The whole point of transcription is to make messenger RNA. So transcription, an mRNA copy of a gene is going to be made from DNA. And what is going to happen is I'm going to try and draw you some really fantastic images of this happening. So this is happening in the no, it's not happening in the cytoplasm, it's happening in the nucleus. So transcription starts, and we're going to have, I'm going to draw for you what it's going to look like to start off with. So, oh, what's happened to my screen here? I don't know what that is. Nope, I did not want to do that. Did not want to do that either. There we go. So here's our DNA. It's badly drawn by myself, but it will do. Now I'm going to explain what is happening on it in a second. T, C, C, T. Right. So transcription starts when an enzyme called RNA polymerase. So we're going to ignore everything else, anything you said, and this is going to be our enzyme. It is really big. It's not tiny. This is our enzyme, RNA polymerase. RNA polymerase attaches to our DNA double helix at the beginning of a gene. So remember, like I said, it attaches at the beginning of the gene. So it's picking that specific region. Attaches at the beginning of the gene, and those hydrogen bonds between the DNA strands in the gene break and separate the strands in that area. Then what happens in that area is that DNA molecule uncoils at that point, exposing some of the bases. Okay, One of the strands is then used as a template to make an RNA copy. So for us, this is going to be, I'm going to use different colour. This one here is going to be our template strand. So if we go through that again, our RNA polymerase has come along. RNA polymerase has come along, and what has happened to it is it's attached at the start codon. It's attached at the gene at the start codon. Okay. RNA polymerase, what it does is it breaks those hydrogen bonds, between the two strands. Our strands unwind. And then one of the strands is used as a template. So this is going to be our template strand here. Now, we'll look into the template strand in a minute because there's some specifics about a template strand. But I don't want to get into the nitty-gritty of what a template strand is at the minute, because I just want to go through the process. But yeah, at the moment, this is what's happening. Step one, and we'll call this one big step one. RNA polymerase attaches at our start codon. This is our gene that's being expressed. It is going to break our hydrogen bonds, and those two strands unwind in that region, and one strand is going to be used as a template strand. The other, we have lots of different words we can use for these strands. So template strand can also be called, I'm just going to put this here, the antisense strand. Our other strand, and this is where it gets a little bit complicated, is called our coding strand. Which can also be called our sense strand. Now, I'm going to put our DNA out across here, and I'm going to explain why this one's called the coding strand. A, G, G, A. So basically, we're going to make an mRNA copy of this template strand. Okay, so we're going to use the template strand to make an mRNA copy. What the mRNA copy will be is a copy of this coding strand. So this coding strand is the genetic code. That's the side with the genetic information on. Basically, the template strand acts as sort of like a negative, as a blueprint to it. So we then use that to make a complementary base parent of the mRNA, and our mRNA will be exactly the same as our coding strand, but minus, if there was a thymine, it will now be uracil. So that's the idea, that's what's happening there. So that's our first part. So our RNA polymerase attaches at our start codon, breaks our hydrogen bonds, and in that region where that gene is, those hydrogen bonds being broken causes our DNA to unwind. So then we can go on to the second step. Let's move that up here, out the way for a moment, and we'll come back to it in a second. Oops. Here we go. So now, template strand. Template strand is going to be transcribed to make mRNA with the same sequence. So let's draw this out now. So again, we've got C, T, C, C, T, and over here, just so I can show you, you've got your G, A, G, G, A. Okay. So this is our template strand again. I'm just going to put a little line to it just to remind us, template. So the RNA polymerase, what that is also able to do, so our RNA polymerase is still here. So I'm just going to pop that back up here. What our RNA polymerase is able to do is it lines up free RNA nucleotides. So like a DNA replication, we have nucleotides, free floating nucleotides inside our nucleus at all times. RNA polymerase is able to line up free RNA nucleotides alongside our exposed base on our template strand. So what will happen is a G will line up here, an A will line up here, a G here, a G here, and an A there. The free bases are attracted to exposed bases, the same as before. Now these are exposed because they're on the outside, and these ones are still on the inside, but these have been exposed to the outside, okay? Specific complementary base pairing means that our mRNA strands end up being complementary of the DNA template strand, okay? So we've got a complementary. So they're going to look very similar to this part over here. And once our RNA nucleotides are paired up with their specific bases on the DNA strand, they're joined together by RNA polymerase, forming an RNA molecule. So they're then going to join up like this. And this is looking bad, but you get the general idea. They're going to join up now. So that is our RNA nucleotides start to join together, and all along here inside our nucleus, we've got some free floating nucleotides, and then they're going to come along when they're needed. I mean, we're not going to have a T, are we? That's silly. And they're going to come along when they're needed. So this is stage 2. So what has happened here? Well, what did we say? We said that the RNA polymerase allows free nucleotides, free RNA, which is important to remember, free RNA nucleotides, and they complementary base pair, complementary base pair with the template strand. The free RNA nucleotides complementary base pair with our template strand. They then join together, and then they join together to form an mRNA molecule. So that is the second stage. First stage, unwinding, hydrogen bonds breaking, unwinding. Second stage, free RNA molecules, nucleotides come along, the complementary base pair with our template strand, and then that molecule joins together to form an mRNA molecule. So, nope, did not want to do that. So our next stage of this, stage 3, let's get that up here now. Let's go underneath actually. Okay, so stage 3. Squiggle some DNA. Yeah, my bad drawing of DNA. That, all right, and we've still got those same bases sticking out here. So what is now happening is the RNA polymerase is actually going to move along the molecule. So obviously it was up near the top originally, so maybe up here, and now it's moved further down. So that's our RNA polymerase. So still got that. That's our RNA polymerase. It's really important to remember. So RNA polymerase moves along our DNA. So it moves along our DNA and it separates the strands and assembles the mRNA strand. So basically our mRNA strand is getting longer and longer and longer as the RNA, oh, it would help if I wrote polymerase in here. Polymerase. So as RNA polymerase moves along the DNA, the mRNA strand forms. So it basically goes through all those first two steps every single time the RNA polymerase molecule moves. So the hydrogen bonds between the uncoiled strands have to reform eventually, basically. So what happens is RNA polymerase moves along the DNA. It separates in the regions where the RNA polymerase is. The strands are separated by breaking those hydrogen bonds, and then we assemble the mRNA strand. But it's important to remember that once the RNA polymerase has moved from that region, so once, and I'm just going to put RNA poly has left that section, our hydrogen bonds, so our H bonds, our hydrogen bonds reform. And they reform once the RNA polymerase is passed, so it can coil back to a double strand, to our helix. So basically, step one is happening here, and then step two has happened up there. They've reformed, okay? Finally, and this is our final bit of transcription. Here we go, and that will be step 4 here. Let's zoom it in a bit. Here we go. Stage 4, our last bit. Here's our DNA molecule. So when our RNA polymerase reaches a particular sequence of DNA called a stop signal, so our RNA polymerase will reach a stop signal. When it reaches this stop signal, it stops making our mRNA and it detaches. So that's the main part, it detaches once it reaches that stop signal. And then what happens is our mRNA molecule moves from the nucleus through a nuclear pore and attaches to a ribosome. And this would be our mRNA molecule up here. So I'm going to draw it on from earlier. So we've got C, G, A, U, C, A, G, A, G. So RNA polymerase reaches a stop signal, detaches from our DNA, and our mRNA moves from the nucleus through a nuclear pore and attaches to a ribosome. So I said something about the different strands. So let's just have a quick look at this as an overview. first before we do anything. So we can sort of basically split up into 4 parts. So this is all transcription. And I can send you this in a bit if you want. So basically, this is all transcription. First stage of transcription, RNA polymerase comes along. It attaches to a gene, and it attaches to the gene at something we call a start codon. Where it attaches, it breaks those hydrogen bonds between the 2 strands of DNA, and those strands unwind. That leaves something called a template strand and a coding strand available. Template strands run in a specific direction. They run in a direction that is called 5' to 3', and we'll look into that later. Okay, so it works 5' to 3'. But don't worry about that too much at the moment. So that's what happens there. RNA polymerase arrives, attaches at a start codon, it breaks those hydrogen bonds there, and the DNA starts to unwind. We've then got a coding strand and a template strand. The template strand is what is going to be used by the RNA to complementary base pair to make the mRNA molecule. So let's have a look how we start making that mRNA molecule. That's step 2. Well, RNA polymerase, while it's in this region, allows free nucleotides to complementary base pair. I did want to do that. Didn't want to do that. Allows it to complementary base pair with the template strand. So we've got all those free nucleotides floating around in our cytoplasm, in our nucleus. G is going to pair with C, A to T, C, G to C, G to C, A to T. Now remembering, if there was a T on that template strand, it would be a U that joins because we have uracil in RNA, not thymine. Once we've got those base pairs there, the strands start to join together to form that mRNA molecule. So that's our mRNA molecule started to form. Our gene is going to be bigger than the RNA polymerases. So our RNA polymerase are going to move along our DNA strand. Every time it moves along the DNA strand, those hydrogen bonds are going to break. The DNA is going to unwind. It's going to complementary, free RNA nucleotides are going to complementary base pair with the template strand. Once RNA polymerase leaves a region, the hydrogen bonds are reformed and they coil back to form that helix. Now, what is really important is I'm just going to go back to this image here. The mRNA molecule here is identical to this strand, which we call the coding strand. Obviously, if there's a uracil there, it'd be identical, but obviously we've got the change of the T and the U. The coding strand is where the genetic information is. If the RNA came along and complementary base paired to this, we wouldn't have, oops. We wouldn't have an identical copy of our coding strand. We'd have the reverse, we'd have the opposite, we'd have the complementary. The triplet codons are what we get off our coding strand. Therefore, we need our mRNA triplet codons to match our coding strand. So they complementary base pair off the template strand. So that's really, really important to remember. So that is why they are identical, our mRNA strand, apart from the difference of uracil and thymine. Our mRNA molecule, so that's our mRNA here, will be identical to our coding strand. So if we go back to here, mRNA strands forming. Every time the RNA polymerase leaves a region, hydrogen bonds reform, and they coil back to form that double helix. And then finally, Once RNA polymerase reaches a stop signal, it will detach from the DNA. Our mRNA molecule then will move from the nucleus via a nuclear pore and attaches itself to a ribosome. And that is transcription. I'm just going to get another image of transcription to quickly go through it. Let's just make this a bit smaller. So yeah, so they're basically the four main steps of transcription. Let's open this up. Okay, so what we've got here is another overview of transcription. And that is what's going to happen there. We've got the mRNA being formed off that DNA. So if we just go back to here, let me change my screen sharing. You can see what I'm about to show you. Share. So how, this is the last little bit I just want to talk about before we finish for today. So how is transcription actually initiated? So I've said, oh, it goes to the site start region. So in eukaryotic cells, before we can actually start transcription, we need a gene to be stimulated by a regulatory protein, and we call this a transcriptional factor. And transcriptional factors contain a few things, and they contain basically these sites that can bind to a specific region of DNA. They cannot initiate transcription alone, but basically what they do is they form a pre-initiation complex with RNA polymerase. So it's actually quite complicated. But basically we have a transcriptional factor and they're in these promoter regions. So when we say specific region, they're these promoter regions and they form a pre-initiation complex with RNA polymerase. And they work in different ways, and that's something we'll look at in the future. But there's a couple of different ways they can work. Some can recognize parts of a promoter sequence, which is that region at the beginning of a gene, and bind to them. And they can either promote or block the function of RNA polymerase. So this is how we can switch genes on and off. So that's what transcriptional factors are. That's how we switch a gene on and off. So they can either promote RNA attaching and working, or they can block it. So the action of a transcriptional factor can be switched off by inhibitors or switched on. And I'm not going to go into any more detail than that in a minute. So as a quick overview, let's go through this. What is a sequence of events during transcription? So in eukaryotes, the transcription of a gene is activated by a transcriptional factor. This moves into the nucleus and it binds to a specific region of DNA. Now, this is going to be the enzyme helicase because it does. The enzyme helicase breaks the hydrogen bonds between the bases in a specific region of double-stranded DNA. You don't have to say that for AQA. For AQA, you only have to say RNA polymerase. A couple of other examples, like not Edexcel, OCR, you have to say that it's helicase and RNA polymerase. But if you say both, you won't lose any marks. So that's why I'm sort of including both. So the enzyme helicase breaks the hydrogen bonds between the bases in a specific region of double-stranded DNA. And then the enzyme RNA polymerase binds to the promoter region. It moves along the single strand of DNA, forming a strand of mRNA, from complementary RNA nucleotides. Transcription starts when the terminator region is reached. Go to stop region. Okay, so that is transcription in sort of a nutshell. What we'll have a look at tomorrow is a little look at bases and the codons again. So actually how these codes work, a bit of a quick overview of codons again. And then look at how we translate them. How do we actually, okay, we've got this mRNA molecule, but what do we do with that now? So we'll look at that tomorrow and then maybe do an overview on Thursday. So fab. I will pop this on to YouTube in a bit.